Hi everyone, we are Team 13 and today we will be discussing the Western blot data from a study of human islet response to type 1 diabetes associated bacteria by Abdelatif and others. First off, the authors effectively communicate the study rationale. Recent research has reported that intestinal dysbiosis is commonly found in newly diagnosed type 1 diabetes patients and is associated with the presence of inflammatory cytokines. These studies consistently indicate that bacteroids, including Bacteroides doré, BD, and Ruminococcus navis, RG, are overrepresented in the gut of susceptible individuals. These same individuals were also found to have increased intestinal permeability. This begs the question of whether bacterial invasion and gut dysbiosis play a role in the pathogenesis of type 1 diabetes through inflammatory pathways.
To answer this question, pancreatic islet samples were isolated and treated with IL-1 beta, RG, BD, or E. coli. RNA sequencing was used to identify NAMPT, SOD2, and ACT1, which are three upregulated transcription factors observed across all bacterial treatments. A Western blot was used to confirm the presence of these inflammation factors for bacterial invasion and gut dysbiosis. The Western blot methods were exhaustive, allowing for high reproducibility. An image of the blot is presented in Figure 6a, where we see the expression of the three factors in GAPDH when treated with IL-1 beta, E. coli, and a serum-free control. We see that the inflammation protein bands are much more prominent when compared to GAPDH, indicating increased protein expression. Figures 6b to d present the relative expression of each inflammation protein to GAPDH under each condition. In accordance with the blot image, relative expression was significantly higher in both treated samples for each protein. Overall, the results are presented clearly, but it becomes difficult to discern important differences because of the lack of p-values and significance indicators. The original figure legend also didn't include abbreviation definitions, making it difficult for the figure to stand alone.
These findings indicate that bacterial invasion plays a role in type 1 diabetes pathogenesis through activation of islet antimicrobial response, because upregulated expression of all three factors is thought to activate signaling pathways that lead to islet inflammation and autoimmunity. But the small sample size of 8 healthy donors limits the strength of the conclusions drawn. So future studies investigating the effects of these bacteria isolated from diabetic rather than healthy individuals is needed to validate these results, as they could induce a different islet response. Since we know that type 1 diabetes currently has no cure, confirming the role of gut dysbiosis in its pathogenesis is exciting, as it may lead to developing powerful prognostic and therapeutic tools in the future.